Okay, so we just finished off with the food entering the stomach, leaving the esophagus, and the process of deglutition. We're in chapter 14. We are going to continue with a condition known as GERD, which stands for gastrointestinal or gastroesophageal rather reflux disease. This is when that lower esophageal sphincter does not close completely, and the acidic contents of the stomach back up into the lower esophagus. The result is irritation of the mucosal lining of the esophagus. This gives you that heartburn feeling. GERD is actually a chronic condition that can lead to permanent damage of the esophageal lining. The treatment for GERD includes avoiding food that stimulates stomach acid secretion, acid neutralization with antacids, and there are special drugs to inhibit stomach acid secretion as well. So let's talk about the stomach and the digestive events that take place in the stomach. Eating takes place at a faster rate than digestion. So the process of chewing your food and swallowing, sometimes we do it so quickly that we look down and we say, whoa, what happened to all of that food? Well, the actual process of chemical digestion takes much, much longer. The stomach doesn't only digest, but it also serves as a storage site for that food that has been ingested, but your body just isn't ready to chemically break it down yet. The stomach has rugae inside of it. The rugae are folds of the mucosa that can expand to accommodate food volume. So this is why after a really big meal, your stomach really does feel bigger because it is. The inside of your stomach is very wrinkly with those rugae folds, and then as your stomach expands with the food, it can hold up to 2 liters of food actually. As it expands, those rugae flatten out. There's a pylorus region of your stomach, and that's the site where most food digestion takes place. This is kind of the pit of the stomach, or the bottom of the stomach. Let's see if there's a picture. This doesn't really show it very well. I'll show it in the next picture, but as this bolus comes down, the food isn't going to hang out up here. It's going to plop down into this bottom area, which you don't see on this picture, but that's called the pylorus region. So, oh, there we go. Sorry, I forgot this was the next slide. There's that pylorus region. That food plops in, plop right here. You can see all of that rugae, all of those extra folds, and you can imagine how those extra folds would kind of flatten out as your stomach gets bigger. All of this smooth muscle would actually begin to relax a little bit to allow for the expansion. Now notice there's actually 3 layers of smooth muscle on the stomach. There's an oblique layer, there's a circular layer, and a longitudinal layer. On most of the digestive tract, we only have 2 layers, the circular and longitudinal layer. The stomach has an oblique layer to really allow for lots and lots of churning and lots of mixing. The top part of the stomach is called the fundus. You see the body of the stomach here. This is, again, the area that will fill up first after the pylorus region has been filled. And then that stomach connects to the duodenum, which is the first portion of our small intestine, via another sphincter muscle, another round muscle called the pyloric sphincter. Again, the stomach can hold about 2 liters worth of food. So if you think about a 2-liter bottle of soda, and imagine that it's all crushed up and ground up food mixed with all of your saliva, that's how much food your stomach can hold, give or take a little bit. Now, as it's being digested and churned up in your stomach, very slowly this sphincter muscle is opening, allowing, kind of like an usher almost. Think about going to church, and there's, lots of people trying to leave at one time, and so the ushers will tell you which row can get up next. Or think about a bus station or a bus stop only letting a few people off at one time. It's kind of what this pyloric sphincter does. It only lets a little bit of that digestive food out at a time, so the rest of it's going to remain backed up here, which is why after you eat a really large meal, sometimes it takes a little bit for your stomach to go back down and for you to not feel quite as full. So let's talk about digestion in the stomach. There's a thick mucosa that has lots of gastric pits inside of it. Those gastric pits contain secretory cells. So in other words, if we were to zoom in here to the rugae, you would see lots and lots of dimples or pits which contain those secretory cells. Those secretory cells are releasing gastric juices, including mucus, hydrochloric acid, and various digestive enzymes. digestive enzymes in the stomach are mostly geared towards digesting proteins. There's also, or these secretions, I should say, are stimulated by the presence of food. They're also stimulated by just thinking about food, which is why sometimes if your stomach is empty and it's been a while since you ate, if you sit here and you start thinking about your favorite meal, maybe a steak dinner or maybe some pizza or lobster or whatever, your stomach will actually begin to secrete some of these substances in preparation for digesting them later. There's also peristalsis and churning of the stomach, which is going to mix those digestive enzymes with that chewed up ball of food that we call a bolus, and that's turning that bolus into a substance called chyme, C-H-Y-M-E. It's pronounced chyme. Chyme is a thick, kind of sticky liquid of digestive food. This animation here goes through the process of stomach peristalsis. This animation goes through the chemical digestion using gastric acid. Let's talk about some of those chemicals in the stomach. I mentioned the digestive enzymes. Pepsin is an enzyme that is actually activated by the presence of hydrochloric acid. It helps to break down proteins into smaller peptides. There's also gastric lipase, which will break down lipids. There's gastrin, which is not an enzyme but a hormone. It's a hormone that's secreted into the blood to stimulate the secretion of gastric juice, stimulate the motility within the GI tract, as well as to stimulate that pyloric sphincter to opening to allow the food from the stomach to get into the small intestine. The stomach is also going to absorb special substances, as I mentioned, mostly fatty substances, some drugs like alcohol. The gastric pits can also absorb water and ions. Here you can see what I mean by gastric pits. So you have this inner lining, the mucosa, and it's all folded up because that's the ruggy. And then inside the ruggy are lots and lots of pits, these gastric pits, which go way down. They have different types of cells lining them. You can see some of these different color cells. Those different cells make up various glands, like a gastric gland, which may be secreting hydrochloric acid or pepsin, for example. In that submucosal layer, you have your lymphatic nodules and connective tissue, as well as a small muscular layer. Then you have your muscularis layer, which contains the longitudinal, the oblique, and the circular layers of muscle. And then the serosa, which is just this outer skin of the digestive tract, just that outer skin. Here you can see the anatomy of the gastric pit. So you have special surface mucus cells to secrete mucus on the surface of our stomach. Then there's mucus neck cells, which are secreting mucus within the neck of the pit. There's special cells called parietal cells, which secrete hydrochloric acid and something called an intrinsic factor, which is essential in absorbing vitamin D. There's chief cells, these little purple guys. Chief cells secrete pepsinogen and gastric lipase, and then the G cells secrete gastrin, which is that hormone to stimulate the motility of the GI tract and the relaxation of that pyloric sphincter. This animation here goes through the process of protein digestion in the stomach. This animation goes through lipid digestion in the stomach. So let's talk about why the hydrochloric acid doesn't eat a hole in the stomach. And that has to do with, let me go back just briefly, these special mucus cells which secrete lots of mucus on the surface of our, on that mucosal lining of our stomach. So HCl secretion is only going to occur when food is present in the stomach. Your stomach does not secrete HCl all of the time. It's only in the presence of food. The chyme, or the food that you bring in, is going to dilute that acid, so even though the acid is very, very acidic, once it is secreted, it's absorbed by that chyme, which is going to dilute the acid and bring it more to a more neutral pH. Plus, you have that thick layer of mucus covering that mucosal lining. So, now that we're done with the stomach contents are then going to be transferred into the small intestine. Small bursts of chyme are propelled through using peristalsis by that pyloric sphincter. It takes somewhere between 1 to 2 hours after a meal, again depending on how big the meal is. Fatty rich chyme is going to remain in the stomach longer, followed by protein, and then carbohydrates. So carbohydrates are the first thing to leave the stomach, followed by the protein, followed by the fatty components of the chyme. Which is why if you eat a high carbohydrate meal, you may be hungry just a few hours later. Let's talk about the anatomy of the small intestine. So the small intestine is made of 3 parts. Altogether, it's about 5 meters long. So this is actually the longest segment of our GI tract. If you remember, our GI tract is somewhere between 5 to 7 meters long. So the majority of it is made of small intestine. The small intestine is called the small intestine, not because it's shorter, but because its diameter is smaller than the large intestine. So the duodenum, or duodenum, depending on how you want to say it, tomato, tomato, that's the first part of the small intestine. That's where digestive begins. This is actually the shortest part of our small intestine. It's really just that little curve at the very beginning of our intestine. Then there's the jejunum. This is the middle portion, 2 to 2.5 meters long. This is where the majority of absorption is going to occur. The duodenum is where lots of pancreatic secretions, liver secretions are going to come in. The jejunum is where lots of nutrient absorption is going to occur. And the ileum is the last portion before the small intestine meets up with the large intestine. Notice this is a cross-section of the mucosa of the small intestine. This is the jejunum specifically. Look at all of these folds. Lots and lots and lots of folds to increase the surface area. With increased surface area, you have an increased rate of absorption. So lots and lots of nutrients can be absorbed from your food. The small intestine has the same exact layers as the rest of the GI tract, but because that inner surface is highly folded, we subdivide it into 2 categories. The macroscopic folds, or the circular folds, and then the microscopic folds are the villi and microvilli. So here you can see the circular folds, but if we were to zoom in even more, you would see that each of these folds has little microscopic folds, and the microscopic folds on top of the microscopic folds, again, just to increase that surface area. Here you can see what I mean. So these are the villi that are on our mucosal lining of our small intestine, and these villi would be folded up around each other within those larger circular folds. If we were to look at each one of these little cells, you would see that each one of these cells themselves also have lots of microvilli on top of them to increase the surface area of each individual cell. Within the villi, within these large projections here, to increase the surface area, you have arteries and veins to, again, absorb all of the nutrients from the cells, make sure these cells have lots of oxygen, get rid of waste products. There's also a special lymph vessel called a lacteal. The lacteals fill in these villi to help suck up lots of the lipids from our digestive tract and take them to the lymph nodes. So the lacteals also serve to help with the pathogen invasion and kind of immune response that needs to take place at this level. As I mentioned, if you were to look at each individual cell on these villi, you would see that they have microvilli on top of them. Microvilli is different than cilia. Cilia are hairs. Microvilli are actually extensions of the cytoplasm to help ensure that that cell can absorb lots and lots of nutrients. Within the small intestine, there's also goblet cells, which are single-celled exocrine glands, basically single-celled glands to secrete mucus. There's also enteroendocrine cells, which secrete hormones like secretin, cholecystokinin, or CCK, as well as GIP, which is called glucose-dependent insulinotropic peptide. So the secretin is going to stimulate the secretion of those pancreatic juices, which are lipases and proteases and DNases, to help break down various molecules for digestion. CCK is also going to stimulate the pancreatic juices. It's also going to cause ejection of bile from the gallbladder. It's going to inhibit gastric emptying, and it's going to induce that satiety, that feeling of fullness and satisfaction. The GIP is going to stimulate the release of insulin from those beta cells of the pancreas. There's also Panin cells. These are special cells to secrete lysozymes to aid in immune response and phagocytosis. They have that lysozyme enzyme to kill bacteria. So let's talk about those villi. They are projections of the mucosal cells that contain special connective tissues, the blood vessels. Whoops, my antivirus is now available, apparently. Contain those blood vessels, as I mentioned, as well as those lymphatic vessels called lacteals to aid in absorption, diffusion, as well as passive and active transport of sugars, amino acids, and lipids. Those villi are lined with special absorptive cells, secretory cells, as well as endocrine cells that I mentioned on the last slide. Ultimately, the villi are there to increase the surface area by 300 to 400%, all while maintaining the outer surface for the organ. So the organ itself looks the same, but it has lots and lots of inner surface area for absorption. The villi are going to assist with the removal of sugars, amino acids, and lipids from the lumen, as well as to allow for water to be absorbed by osmosis. Vitamins are also absorbed by simple diffusion, either with water or with lipids, depending on if the vitamin is lipid or water soluble. In between the villi are special intestinal glands to secrete intestinal juices that have water, mucus, as well as enzymes inside of them. So here you can see the absorption of nutrients by the small intestine. You can see this is the actual lumen of the intestine, so food is passing through. These are the special cells of our small intestine, and then this is that submucosal layer of our small intestine that has the blood vessels and the lacteals. So most of our carbohydrates are going to get into our intestinal cells and into the submucosa and into the bloodstream via facilitated diffusion because they're such large molecules. In some cases, they might need to be co-transported with sodium. So either way, they're pumped out into the submucosal lining. Sorry, I lost my train of thought. Into the submucosal lining of our small intestines to go into the blood. Things like proteins are also going to use active transport because proteins are polar molecules. They're going to need to be co-transported sometimes with sodium, as you see here. They can leave via diffusion and go right into the bloodstream. Whereas our fatty acids are going to be broken down by bile salts, the special salts within bile that's secreted by the gallbladder, made by the liver, secreted by the gallbladder. Those bile salts are going to break apart large lipid droplets into smaller droplets called micelles. These micelles are then broken into fatty acids and monoglycerides, which can just be absorbed via diffusion into our cell. The cell is going to convert them into something called a chylomicron, which is kind of like a emulsified fat droplet, or a fat droplet that's kind of surrounded by a bubble. Those chylomicrons are then absorbed by the lacteals, absorbed by the lymphatic vessels. Then they circulate in the lymph and return back to circulation. Here you can see at the heart, just like all of our circulation returns. Something interesting, if you check out this this pathway here, you can see all of these blood vessels eventually, of course, going to lead back to what's called the hepatic portal vein. So this artery leads to capillaries, leads to venules, leads to a vein. That vein, my goodness, my computer's going crazy, sorry. That vein from our small intestines goes to what's called the hepatic portal vein. In fact, all of our small and large intestine veins are going to bypass all the other structures and go straight to the liver. The liver is a big filtering station. It's going to go through, grab any extra nutrients. It's going to secrete extra vitamins and enzymes or whatever else your blood may need before that blood gets to the heart. So let's talk about what's going on in specific areas of our small intestine. The digestive functions of our duodenum include the acidic chyme. That's going to stimulate those S cells to produce the secretin, S cells, secretin. That secretin is going to stimulate the pancreas to secrete bicarbonate into the duodenum. It's also going to, that pancreatic juice is going to neutralize the acidic chyme. So bicarbonate, you may remember, acts as a base. or a neutralizer, so that chyme that's coming out of the stomach, which has a very low pH, is going to be neutralized by that bicarbonate from the pancreas. There's also amino acids and fatty acids in the chyme that are going to stimulate the CCK cells to secrete the cholecystokinin, the CCK. CCK is going to increase pancreatic secretions for breaking down lipids and proteins. It's also going to cause bile to be released from the gallbladder so those lipids can be absorbed. And it's going to inhibit gastric emptying so that you feel full and so that it gives your small intestine time to absorb the nutrients from the food that's already there. This animation goes through carbohydrate digestion. This animation goes through carbohydrate absorption. This animation goes through protein digestion. This animation goes through protein absorption. This animation goes through nucleic acid digestion, which maybe you didn't think about before. This one talks about nucleic acid absorption. Finally, lipid digestion using bile salts and pancreatic lipase, and bile absorption. There are specific hormones that control digestion. We've mentioned a few of these already. The hormone gastrin, produced by the stomach mucosa, that's going to respond to stretching of the stomach to stimulate the secretion of gastric juices, increase motility, and relax that pyloric sphincter, allowing the food to pass into the duodenum. Secretin, which is going to stimulate bicarbonate ions to come from the pancreas to help neutralize that chyme. And then the CCK, coming from the duodenum mostly, that's going to be stimulated by amino acids and fatty acids in the chyme to inhibit gastric emptying, to stimulate pancreatic-rich enzymes to be released, as well as the ejection of bile from the gallbladder. This animation here goes through hormonal control of digestive activities. So let's talk about the special movements in our small intestine. There's actually two types of movements. You've probably heard of peristalsis already. That's when that chyme is just moving along down the length of the intestine, and you can imagine various substances being absorbed at various stops throughout the small intestine. But there's also a process called segmentation. Segmentation mixes the secretions. So segmentation is kind of when forward movement stops and just a general localized churning takes place at that one segment, hence segmentation, of the intestine. So in this case, segmentation is just going to mix the secretions with the chyme, mix the enzymes with the chyme, get them churned up, and then peristalsis moves it through. This is an animation of segmentation. Let's talk about the liver secretions. The hepatocytes, which are the specialized liver cells, make bile. Bile contains bile salts. Bile salts emulsify or break down large lipid droplets into smaller droplets called micelles. Think about this. You're probably aware of it. If you have a really greasy pan and you're trying to wash it with just water, is it going to become degreased? No. You have to use soap. Soap is an emulsifier, just like bile is an emulsifier. So as you use that Dawn or Palmolive or whatever to scrub that greasy pan, all of those fat droplets or oil droplets on your pan are emulsifying and breaking down into smaller droplets. That's the same idea as when you're eating something fatty and bile is mixing with it. The pigment in the bile is mainly bilirubin, which is derived from worn out red blood cells. It's that kind of yellowish, greenish color that's derived from that broken down hemoglobin. The liver helps with many processes dealing with the absorbed nutrients from digestion. There's that hepatic portal vein that I showed you. That's going to transport the nutrient-rich venous blood to the liver. That blood is then distributed to the hepatocytes through special capillaries called sinusoids, and those hepatocytes are going to pull out any excess nutrients, processed nutrients, as well as secrete any various vitamins or enzymes into the venous circulation. Those hepatocytes combine simple carbohydrates like glucose to form glycogen. They also help to create lipids from glucose and amino acids. So, in other words, if you have too much glucose floating around in your blood and you're not using it for energy, your liver is going to be the guy to convert it into fat. It's also going to help distribute amino acids to the blood, so making sure that your blood has high enough protein levels and amino acid levels. It's going to help to break down excessive amino acids as well as worn out proteins into glucose or triglycerides. After the hepatocytes complete their work, the blood is going to drain from those hepatic sinusoids to central veins, to the hepatic vein, eventually to the inferior vena cava, up to the right atrium, and so on and so forth. This animation here goes through the chemical digestion use of bile. Since we were just talking about the liver, let's go ahead and talk about the gallbladder. The gallbladder stores bile. Bile is made in the liver, is stored in the gallbladder. CCK is going to trigger that release of bile into the duodenum through a duct called the common bile duct. The pancreas is another really important accessory organ when we talk about digestion. The pancreatic juice is produced by the acinar cells of the pancreas, as opposed to the islet cells like the insulin and glucagon are. It's clear and colorless. It flows into the duodenum through a duct called the pancreatic duct. It's composed mostly of water, sodium bicarbonate to help neutralize the stomach acid, as well as special enzymes like pancreatic amylase, trypsin, chymotrypsin, carboxypeptidase, pancreatic lipase, ribonuclease, and deoxyribonuclease. The pancreatic amylase is going to continue with carbohydrate digestion. Trypsin, chymotrypsin, carboxypeptidase, those are all for protein digestion. Lipase is for lipid digestion. Ribonuclease and deoxyribonuclease are for nucleic acid digestion. And this is pretty much what I just said. I forgot this slide was here. Amylase is breaking down the starches into sugars. Trypsin, chymotrypsin, carboxypeptidase, breaking down those proteins into amino acids. Lipase is breaking triglycerides into fatty acids. And then the nucleases are breaking down nucleic acids. Here you can see the anatomy, configuration of the liver, the pancreas, and the gallbladder. So you can see how intimately all of these things are combined. This is the duodenum, that first segment of the small intestine. The stomach would normally be sitting right here. You don't see it in this case. You can see the pancreatic duct and the common bile duct coming down from the liver, dumping into the duodenum. They're secretions, and those secretions would mix with the chyme. Of course, the chyme's coming from the stomach right here, dumping into the duodenum, and then going to be passed through to the jejunum with segmentation and peristalsis. So let's talk about, let's kind of give a summary to the function of the small intestine. There's segmentation, that's mixing the chyme with digestive enzymes, ultimately bringing food in contact with the mucosa for absorption. There's peristalsis, to push the chyme through the small intestine. The small intestine is completing the digestion of carbohydrates, which actually began in the mouth. Proteins, which protein digestion began in the stomach. And lipids, and lipid digestion also began in the stomach. It begins and completes the digestion of nucleic acids with those nucleases, and it absorbs 90% of nutrients and water that go through the digestive system. So the majority of the absorption occurs in the small intestine. Where do the absorbed nutrients go? Well, amino acids, sugars, fatty acids, they're all going to go from the villi epithelial cells into the hepatic portal circulation through diffusion to the liver. They're going to be processed in the liver and then put back into the general circulation. So let's talk about the nutrients go after the small intestine. Fats are going to be packaged into those chylomicrons, those little spheres of fat. They're going to leave those absorptive cells through exocytosis, be absorbed by those lymphatic vessels called lacteals. Those chylomicrons are going to travel through the lymphatic system, get into the general circulation, eventually to the adipose tissue and to the liver. At the liver, the chylomicrons are removed and lipids can be stored in triglyceride form and deposited in your subcutaneous layer or within your greater omentum. When the chyme reaches the large intestine, the bile salts have been reabsorbed and returned to the liver for recycling. Insufficient bile salts can result in a 40% loss of dietary lipids in your feces. So here you see this animation again, or this picture again. We have the idea of carbohydrates being absorbed through facilitated or active transport, leaving these absorptive cells going into the circulation, as well as proteins being absorbed through active transport, diffusing out, going into the circulation. This circulation is going to go through the hepatic portal vein, through all the hepatocytes to be modified or absorbed. Then all of that blood is returned to the inferior vena cava, goes back to the heart. The lacteals, same idea. Lacteals are going to absorb those chylomicrons. Chylomicrons are going to be transported via the lymph to the liver. Within the liver, really, they're going to be transported back to the heart and go through the general circulation, where they eventually will come in contact with the liver, who will then remove the chylomicrons and store the lipids if they're not absorbed for energy purposes or metabolic purposes by cells. The liver will store those fatty acids for later use. So now we get to the last stop, kind of, on our tour. This is the large intestine. The large intestine is called the large intestine because it has a larger diameter. It's about 1.5 meters long, so we're talking about 4 feet, 5 feet or so. It extends from the ileum, which is, of course, the end of our small intestine, all the way to the anus. It's attached to the posterior abdominal wall by a mesentery called the mesocolon. It has 4 segments: the cecum, the colon, the rectum, and the anal canal. The ileocecal valve is going to regulate the movement of chyme into the large intestine. Ileo referring to the ileum on the small intestine. Cecal referring to the cecum of the large intestine. The internal and external anal sphincters are going to regulate the movement of materials out of the large intestine and into the world, or hopefully the toilet. There's also structures called tentacoli. These are just bands of muscle fibers. This is what causes that puckering appearance of our small intestine, which forms these little pouch-like sacs. The pouch-like sacs are called haustra. They increase the surface area for absorption of water, sodium, potassium, vitamins B, and vitamin K. Here you can see those special bands. I'm trying to see where they're labeled here. I don't see the tentacoli. Oh, here we go. The tentacoli makes up this large kind of tract here, and then it forms little bands around the small intestine, which forms the pouches called the haustra. This first little pouch coming from the ileum, here's the ileum, you can see how narrow that small intestine is compared to the very wide large intestine. The cecum is kind of like the catcher's mitt in this case. Coming off of the cecum is the appendix. You can see that here. So you have the cecum, your ileocecal sphincter controlling the movement from the small to the large intestine. Then the colon itself, which is consisting of the ascending colon, the transverse colon, the descending colon, and then the sigmoid colon, which is this weird kind of S-shaped. Then you get to the rectum and the anal canal, and finally to the anus. If we were to zoom into the anus, which is kind of an odd thing to say, you can see the rectum and then the anal canal being this very last segment. The anal canal itself has 2 sphincters, an involuntary internal sphincter, and then an external sphincter, which is voluntary. So children do not yet have control, I should say non-potty trained children, do not yet have control of their external anal sphincter, which is why they poop all over the place or can't tell you necessarily when they have to poop. It's when they are finally potty trained that they have control over that external anal sphincter. If we look at the mucosal lining of the large intestine, it's very similar to the other setups. You have a smooth mucosal lining here. There's not very many folds for increased surface area. There's no villi. It's not the same exact setup as what we had in our small intestine and stomach, but there are pits. Our cells do have microvilli on them to help absorb lots of water. We want to make sure that the large intestines have absorbed as much water as they possibly can. You still have the same setup within that submucosa of the blood vessels and the lacteals and the lymphatic nodules. There's still some special mucus secreting cells called goblet cells in there as well. Overall, the functions of the large intestine include churning, peristalsis, mass peristalsis, which is when the colon kind of has a huge lumbering contraction to move the contents into the rectum, and that's when you say, ooh, I gotta go to the bathroom. There's also the absorption of water, ions, and vitamins, ultimately forming the feces, condensing that chyme down into a more solid material for feces, and of course it is involved in defecation. So there are special bacteria involved in our large intestine. The function of these bacteria are to ferment the carbohydrates to release gases. The majority of the gases are methane, carbon dioxide, as well as some hydrogen in those gases. It's called flatus, right? Flatulence, you may have heard of before. The bacteria are going to break down proteins as well as amino acids. They're also going to help convert the bilirubin into more simpler pigments called stercobilin. That's going to give the feces its brown color. It's also going to, these bacteria are also going to help produce essential vitamins B and K, which are then absorbed by the colon and secreted into the blood circulation. So what else happens in the large intestines? Basically, any nutrients that are absorbed are going to be passed out in the feces. Absorption of nutrients does not occur in the large intestine. Only water and vitamins are going to be reabsorbed. Basically, 9% of what's taken into the digestive tract is then removed by the large intestine. Only about 1% is lost in feces. If you're having some type of digestive issue, that 1% may be higher, in which case you would have a more watery stool, aka diarrhea. This animation goes through the mechanical digestion process of the large intestine. Anything else happening in the large intestine? Well, the chyme generally takes 3 to 10 hours to pass through the large intestine, and the water is getting absorbed, so the chyme is going to go from a more liquid to a more semi-solid or solid mass that we know as feces. The composition of feces, it's made of water, inorganic salts, any epithelial cells that may have sloughed off that mucosal lining, lots of dead bacteria, the products of the bacterial decomposition, any digestive material that was not absorbed, and any indigestible parts of food. Defecation itself begins with those waves of mass peristalsis that occur periodically. Generally, they occur twice a day, and sometimes you might not feel the urge, and then maybe the next time you will feel the urge. The urge to go is what I'm referring to. This is ultimately driving feces from the colon into the rectum, initiating the reflex that we call the defecation reflex. Let's talk about the defecation reflex. It involves special stretch receptors within the walls of the rectum. It's under parasympathetic nerve activity to relax the anal sphincters. Sometimes the diaphragm and abdominal muscles can aid in initiating defecation, and you know that if you've ever held your breath and tried to squeeze it out a little bit, kind of silly to talk about. But there's also contraction of the muscles within the anus to stop the feces out. That would be the voluntary contraction, saying, "Oh, no, don't go now. I'm in the middle of lecture, or I'm, in the middle of a date or something, and you don't want to go at that time." However, once in the rectum, the feces will then exit involuntarily through muscle contraction. So it's that voluntary external sphincter that keeps it in, and then once you sit down and let go of that voluntary muscle, everything else takes place under involuntary control via the defecation reflex. So here you can see, same picture that I showed you before, those mass movements ultimately pushing that food through, pushing that chyme, I should say, through, getting it from this kind of relaxed state here, getting it up and over this hump and then into the rectum, and that's when you say, "Whoa, I gotta go." And your internal external sphincters will remain contracted until you tell them to relax. Am I even recording right now? I am, sorry. I didn't see my little thing. Hang on. Okay, sorry about that. I usually have a little box down here that's telling me if I was recording, and I didn't see it for a second. So let's talk about diarrhea, shall we? Diarrhea is actually classified as the frequent defecation of liquid feces. So this is when water and nutrients are remaining in the feces instead of being absorbed through the large intestine. This can be caused if the chyme moves too quickly. So if there's an increased motility of the intestines because of overstimulation. This could also be in the instance of possibly enzymes that would usually process the food if those enzymes are missing. The absorption of fluid and nutrients by the intestine is going to decrease, so individuals that suffer from frequent bouts of diarrhea could suffer from dehydration as well as malnutrition because the nutrients and water are not being brought into the body like they should be. Constipation is the reverse. This is infrequent or difficult defecation due to decreased motility of the intestines. When the movement of chyme to the intestines is slow, the absorption of fluid increases, which is going to lead to a more dehydrated feces. It's going to be harder to pass, harder to move, as well as harder to squeeze out, if you know what I mean. So all of this is regulated by the enteric nervous system. The ENS, as previously mentioned, that's a gut brain, or the brain of your gut. Those neurons are located within the GI tract themselves. They're influenced under autonomic nervous system. So there's motor and sensory neurons. The enteric nervous system motor neurons control muscle movements and secretions within the gastrointestinal tract. The sensory neurons are going to detect the presence of food through mechanical and chemical stretch receptors within the gastrointestinal organs. So there's 3 phases of digestion that are controlled by the enteric nervous system, and the first phase is called the cephalic phase. Cephalic, you may recall, refers to our head. So this is when just thinking about food is going to get our digestive juices flowing, so to speak. This can respond to the smell of food, the taste of food, as well as the sight of food, just looking at pictures, or as I mentioned, sometimes just thinking about it will get your mouth and stomach ready for the food. That's why if you're very hungry and then all of a sudden you smell popcorn, or you see a commercial for a really good burger or something, your mouth begins to water a little bit. That phase is actually going to last as long as you're eating the meal. So it's going to continue while you're ingesting that entire meal. Then there's the gastric phase. The gastric phase is going to continue those secretions as well as motility. This is when the mixing of food occurs, and this is going to regulate the gastric emptying. So in other words, regulating those spurts through the pyloric sphincter into the duodenum. There's then the intestinal phase. This is going to promote continued digestion through the small intestine. It's going to inhibit gastric emptying. It's going to make you feel fuller. It's going to prevent the duodenum from being overloaded with chyme so that absorption can take place nice and slow like it needs to. This animation goes through the neural regulation of mechanical digestion. Whoops, I think I went backwards. There we go. This animation goes to the enterogastric reflex. And this chart from your book goes through a summary of digestive functions of all of the organs within the gastrointestinal tract. So we'll stop here for part